when we talk about insecticide resistance, there seems to be a bias. We automatically kind of think about malaria, which is fair enough given the high-profile nature of malaria and some of the issues around resistance on that. So we thought we'd rebalance that slightly. And so we've got two wonderful speakers, two wonderful groups actually speaking for this particular session. Joining us all, the Centre of Investigations in Microbiology and Tropical Parasitology, CIMPAT at the Universidad de los Andes in Colombia, Dr. Johan Manuel Calderón. He's going to be speaking about Deltamethrin resistance, but within the setting of Chagas disease vectors and oil palm plantations. So there's some implications there in terms of vector control strategies, public health to agricultural interface, multi-sector approach, as it were. And so that would be very interesting to find out about that. And then we're going to have another talk. We're going to have the second talk. It's going to be in Spanish, and it's going to be translated into English. So you'll hear it in Spanish, and then you'll hear it in English for the second talk. And that's going to be Dr. Rosa, Patricia, Penilla, Navarro, the Instituto Nacional de Salud, Pública at Tapachula, Mexico. It's going to be in Spanish. The translation is going to kindly be handled by Dr. Saúl Lozano from the Centers of Disease Control and Prevention. Very kind of Dr. Lozano to offer to translate that live. It's not an easy thing, but that's going to be looking at insecticide resistance in Aedes aegypti from within that region of Mexico, both spatial variation and response to historical insecticide use. So a very interesting session coming up. So we think we'll kick off with Dr. Johan Manuel Calderón. Johan, the floor is yours, and I'd just like to say welcome and thank you to all the panelists, all the speakers. So over to you, Dr. Calderón.
Okay, thank you for this opportunity. I'm going to talk about one of the chapters of my thesis in my PhD, and I'm going to present you the one problem that we are finding in Colombia that is related with Chagas disease vectors. We have Chagas, the disease is Chagas disease that is very important in the world and in Colombia. In the world, we have 10 million people infected and 25 million people in some risk zones. And specifically for Colombia, we have 500,000 of people infected and probably 30.5 million, probably more people in inhabitants of risk zones for the disease. For that reason, we have a very important disease for Colombia, for South America, and also Central America. The Chagas disease is caused by, is a zoonosis caused by the parasite Trypanosoma cruzi, which is transmitted mainly by these vectors that are the triatomins. The triatomins are usually, they take the blood, and usually when they for losing some weight, they transmit the parasite in their feces. And we have the problem with the disease that we have a very complex transmission cycle. When the problem is we have several hosts for the Trypanosoma cruzi. We have several mammals that could be the host for that. And based on that, we have a problem to eradicate the parasite. This is impossible, I think, because of the big amount of hosts that we have in the transmission cycle. And due to that fact, we have to control the triatomins, or have some way of control or know about the ecology of the triatomin vectors for avoiding the problems of transmission. For that, usually the research has been focused on the vectors, on triatomins. We have the idea is to control the transmission, the main step in the transmission of the parasite between the hosts, usually the wild host, to the human population, usually very close to those places. Between the triatomins usually are very associated to one type of habitat. Usually they prefer places with shelter, with a good place for hiding, and they need also places for or host blood sources for living. Due to that, we need the triatomine usually are very associated with some habitats. One important habitat in Colombia are the ponds. Ponds are very common in Colombia. We have several species of ponds. We have part of the country that are very that have high abundances and diversity of ponds. And with that, we know, for example, that one group, one specialty group, that is the Rhodnius genus, is very associated with those ponds. Why? Because the pond offer shelter and blood, and with those two conditions, we know that the Rhodnius is good with that. That are needed, there are conditions needed for them. And due to that association that is very studied in South America, in Colombia, Venezuela, and Brazil, there have been a lot of different studies about that association. And that the association is very important, for example, in the special case of Colombia, because we have several of big groups of ponds living close to the houses of people. Usually a group of ponds, a cluster of ponds, are usually very located close to the houses, and the Rhodnius prolixus or Rhodnius, several species of the Rhodnius genus could travel or fly to the houses, and the transmission could begin in the house, the transmission of the Trypanosoma cruzi. In Colombia, we have an additional problem. We have a huge and increasing amount of oil palm plantation, because the palm oil is a product very important for exportation in Colombia, and the production, the cultivation and the plantations in Colombia have increased during time. But every monocrop, every big crop usually have some pests, and the palms have several pests, important pests, that usually create or produce several decreasing in the productions, and due to that they use the production or the use of insecticides. We have here two of the most important pests that we have for the oil palms. And the problem is that usually for the control for that pest, usually they use some pyrethroids, like for example cypermethrin, cyalotrin, deltamethrin, and they are also being reported as recommended for the triatomine control. With that similarity, with the use of those pyrethroids for this control, but also for palms, where we know previously my research, the research group where I work with in the CIMPAT has identified several parts of Colombia where we have found triatomines living or inhabiting ponds. And with that, we know that in some parts of Colombia we have triatomines living in those ponds. If we have the problem that the production of the producers of palm oil are using insecticides for controlling the pests, and in the same pond we have also triatomines, the possibility is that the constant exposition to insecticides could affect or could increase or produce insecticide resistance. And that problem is if those triatomines go to houses, and the idea for controlling those triatomines is with the same insecticides that they are using. They are resistant against because they are also being exposed in the ponds. And that is the main problem that we have found, and that is the situation. And my project was to identify, to assess if triatomines inhabiting oil palms exhibit resistance to insecticides used for vector control, is to determine that the possibility of the insecticide resistance. Based on that, we use a Deltamethrin. That is one important insecticide that has been used in Colombia for the control of triatomins. We have our idea with this study was to determine the resistance using two approaches: a biological assays against two presentation of Deltamethrin, pure Deltamethrin, the active principle, and the commercial Deltamethrin in the presentation that is commercial, usually is in a lower concentration. And using also biochemical assays. In those assays, the idea is to see the activity of the toxifying enzymes and see if we see any increase or increment in the activity of those enzymes that could helping to understand the mechanism of insecticide resistance. We made those assays in three populations: in Rhodius prolixus, that is the species more important in Colombia. This is the main vector of Chagas disease in Colombia. And we identified three types of population. Rhodius prolixus collected from oil palms, Rhodius prolixus from a susceptible colony, that is the control that we are going to compare the results between this population against the control, that is a susceptible colony. But we also make an addition and include Rhodius prolixus from a native palms, from palms close to the oil palm crops, but without the exposition of insecticides. And for this population, for us, was a field control. Our idea was to compare those three populations for the resistance and see if we identify any important signal of insecticide resistance. We have found initially with the results of our research that when in the first part that we make assays with pure Deltamethrin, our idea was to test different concentration of Deltamethrin in triatomins and see what and determine or estimate the lethal dosage 50% and 90% and compare those dosage between the three populations. We found that the oil palms, the lethal dosage 50% and 90% were higher than the susceptible. Yes, higher, significantly higher than the susceptible. With that result, we could say that those insects are resistant. But also we found that with native palms, the values are higher than in the susceptible, but for the lethal dosage, 50% are higher, but not significantly. And with the lethal dosage 90%, the dosage for the oil palms population are higher, significantly higher. But with those results, we found that for the pure Deltamethrin, there are a resistance. There was a resistance for that. For the commercial Deltamethrin that is using the commercial presentation, we make a similar bioassays, and we identify that the insecticide, the mortality, in this case is not the dosage letter, but the mortality, using the commercial presentation, we found that the mortality for susceptible were 90%, for native palms was 43%, and for oil palms was much lesser, that was 10%. And we see that with the commercial Deltamethrin, the populations of the triatomin population in oil palms were much more resistant, or the mortality was very lower compared to the other population. But we see that in the native palms, we see any resistance compared to the susceptible. That is usually a triatomin colony in the laboratory or from the laboratory. For the final assay, that is with the enzymes, we see that we compare 4 enzymes. oxidase, alpha esterase, beta esterases, glutathione S-transferases, and the idea was to compare the activity in our three populations, susceptible, native plants, and oil plants, and see a difference in the activity, in the amount of reactant that enzyme could process or could have an activity on that reactant. We see that with we compare with the susceptible, or we see in those box plots the activity, and we see different. You can see different letters that usually it represents a significant difference between those populations, and we see that comparing susceptible with oil palms in the four enzymes, we found higher values of activity compared with the susceptible. Compared with the native plants, we see higher values in some cases, except for the transferases, but we see an activity, a higher activity also in those population from native palms. We have to remember that those native palms usually are very close to the oil palm crops, and probably we have found that there could be any interference or any exposure of insecticide on those plants staying close to the crops. Our conclusion for that study is that Rhodus prolixus for oil palms show resistance against pure deltamethrin and commercial deltamethrin. We have found resistance. Probably our hypothesis, based on what I said in the introduction of this talk, is because we think that the continuous exposition to insecticide for pest control is the cause of that resistance. We see in field that the crops usually are the insecticide that applied using planes or airplanes for if the amount of the area of the crop usually is very big or is huge, they use airplanes for the aspersion of insecticide, and probably we see that, and we think that probably that is the cause, but we have to continue our research for that. And we also think that ponds close to the crops, there is a pond that is very common in Colombia. There is the royal pond. Then usually those cluster of those ponds are very near to the oil crops, and we think that probably the triatomins could fly from, or the population are interconnected between oil ponds and native ponds adjacent or very close to the crops. And we identify resistance lower for pure deltamethrin and for commercial deltamethrin, but probably we think that the main cause is the dispersal between those types of ponds. With that, we found that we think that the possible mechanism for explaining that resistance is the increased activity of the detoxifying enzymes. We think that probably some enzymes have increased their activity, and they are detoxifying the triatomine and avoiding bad damages due to the insecticide. And we have to continue trying to see if we can see other different mechanisms. And finally, for future perspectives about this research, we think that in Colombia, we have, this is the map of Colombia. We see municipalities, that is our regions or the divisions that we have in the country. And we have we have in different colors of green. So the municipalities represented here are municipalities with oil crops, and if you see the color is related with the amount of oil crops inside the municipality. We see that in several municipalities of Colombia, there are oil crops, and in some regions of the country we have huge areas of crops. We also know that we have here these municipalities that have been reported with triatomins that we know inhabits or lives in ponds, and we see that those regions, for example, that are highlighted with the circle, have crops and also have the triatomins. And we see here also the distribution of the municipalities with Chagas disease cases, and the color is related with the amount of cases. We see that inside the circles, we have some municipalities that are important, and we have to be very careful in that places, because we are seeing the possible combination of factors that could give us in the future a big problem with the oil ponds and the transmission. But the idea is to continue with that research. Thanks for your attention.
Dr. Calderón, thank you very, very much, Jose, for that. That was a wonderful presentation and a really interesting interface between this agricultural kind of setting and the prevalence of Chagas. And it's fascinating, the output from your paper. And I wonder, and I'd love to see how that's going to develop moving forward, informing the policymakers, as well as these enzyme assays that probably need to be done as well in terms of the detox pathway within the parasite. I think it'll be fascinating on both fronts. So, you know, really in there, but also at the policy level. I love the fact that it translates to the policy side of things as well. We've had quite a few questions coming through from the audience, which I'm going to channel into the Q&A. So you've really started people thinking. And what I love about we started this kind of online, this is our 8th annual year of this ISNTD Bites, 8th year. And normally it's a meeting where we meet together, everybody meets together in person in London. And we found that because of COVID, a year and a half ago, we started online kind of this approach. And what we found is that you lower immediately the access barriers for people to come. So as an example, from all over the world, so you're working in Colombia right now, you've had your research, we're getting people joining us now. I'm going to give a shout out to the audience as well. So Eduardo Olmedo from Mexico, Bior Garang from the Leprosy and Neglected Tropical Diseases Organization in South Sudan in Africa. We've also got Dr. Alhaji Amadou Niang. He's the assistant professor Alhaji Amadou Niang in the medical entomology division in the Sheikh Anta Diop University in Dakar in Senegal, so Africa. We've also got, and this is in the chat function on the right-hand side of your screen, we've got Daniel Velázquez Ramirez, the PhD student from Mexico, so Latin America there. We have Dr. Helena Uliartha from the NIHRD in National Institute of Health Research and Development in Indonesia joining us. Hi there, Helena. Nice to see you as well. Manuel Espinoza from saying Saludos desde Argentina. So there we go. We've also got Dr. R.T. Rama joining us. He's the Entomological Surveillance Officer for CARE in India. So we're getting north, east, south, west pulling together. And I think your research deserves that, frankly. I think that's a fascinating approach. So, you know, congratulations on that. Well done. Ronald Morales Vargas has joined us as well. A lot of people saying hello. We've got Borel Chamen from the Cameroon, PhD student on Aedes vectors and the control at a center in Cameroon there in Africa. We've got a lot of people joining us, a lot of questions coming through as well. Dr. Celestine Mudugo from the DRC Congo, Kinshasa in Africa. A lot of people, and I really hope, and Mohammed Qasim Khan, I think from Pakistan, is joining us with a few comments and questions that are popular, and I'll channel those into the thing. And what I would say to all the audience is these sessions are designed for everybody to immerse themselves, ask, and we're going to channel that to our wonderful panelists. But I'd like to give you a round of applause, Dr. Calderón. That was a wonderful presentation, and it's elicited a lot of questions, and we'll get to those questions in a minute. Thank you very much for that.
Okay, thank you.
Thank you. And that brings us to the second speaker, the second group. Well, actually, it's a group. We'd contacted Dr. Rosa Patricia Penilla Navarro from the Instituto Nacional de Salud Pública in Tapachula in Mexico. She very kindly put the presentation together. The presentation will be delivered in Spanish by Francisco Solis and then translated by Dr. Saúl Lozano from the Centers for Disease Control and Prevention. He's also at Colorado State University. So he's very kindly going to do, you'll hear it in Spanish and then you'll hear an English translation straight away. They're going to be talking about insecticide resistance, but now outside of Chagas in Aedes aegypti, looking at spatial variation and response to historical insecticide use in that region of Mexico. So over to you. A round of applause for Dr. Calderón. Thank you.
Good morning.
Okay, buenas tardes.
Yeah.
¿Me
We
escuchan?
can hear you. Te podemos escuchar, Francisco.
Buenas tardes.
Good afternoon.
Vamos a presentar nuestro trabajo de investigación que lleva por nombre Resistencia a insecticidas en Aedes aegypti from Tapachula, México.
We will be presenting our research entitled Insecticide Resistance in Aedes aegypti from Tapachula, Mexico.
Variación espacial y respuesta al uso histórico de insecticidas.
Spatial variation and response to historical insecticide use.
Siguiente. Bueno, el dengue sabemos que es una enfermedad que causa miles de muertes en el mundo.
We know that dengue is a disease that causes thousands of deaths in the world.
Aedes aegypti es el principal vector uno de los principales vectores de virus dengue.
And Aedes aegypti is its main vector of the dengue virus.
Okay. En la ciudad de Tapachula, Chiapas, México,
In the city of Tapachula in Mexico,
tenemos dengue endémico con casos todo el año.
we have dengue endemic with cases throughout the year.
También tenemos enfermedades emergentes como es chikungunya y Zika.
We also have emergent diseases like chikungunya and Zika.
El número de casos fueron reportados en el 2015, casos significativos.
There were a significant number of cases reported in 2015 for chikungunya.
Y reportes también en el 2016 de casos de Zika en la ciudad.
And there were also cases of Zika in 2016 in the city.
Todos estos factores.
All of these factors.
Siguiente. Aunado a los factores que tiene la ciudad.
Together with factors that are related in the city.
Como son el clima con lluvia, 6 meses de lluvia y 6 meses seca durante el año.
And the city has a weather with 6 months of rains and 6 months of dry throughout the year.
Lluvias, inundaciones por lluvia.
And there's also floods caused by the rain.
Sitios de deposición.
There's plenty of breeding sites.
Deficientes servicios públicos.
Unreliable water and trash collection.
Y contando también con el comercio y la migración.
And put together trade and migration into the city.
Todos estos factores.
All of these factors.
La siguiente. Todos estos factores son fundamentales para el incremento del número de casos de dengue.
All of these factors are important in the increases of dengue.
Siguiente. Por lo tanto, el control del programa de manejo de vectores.
As a result, the vector control program
intensifica las acciones para contrarrestar las poblaciones de Aedes aegypti.
The program intensifies its efforts to control the populations of Aedes aegypti
Con nebulizaciones espaciales,
with fogging, with spatial fogging,
rociados intradomiciliares con motomochila,
the use of indoor fogging with backpack,
rociados residuales con bombas Hudson,
the use of indoor residual spraying with Hudson pumps.
y todo este uso de control químico
And all of this use of chemical control
genera una presión de selección en las poblaciones del vector.
generates selection pressure in the vector populations.
Lo que hace importante estudiar a la ciudad,
What makes this an important, it is important to study the populations in the city
el comportamiento de la resistencia en las poblaciones.
and the behavior of the resistance in the populations.
Siguiente. Pues como sitio de estudio tenemos la ciudad de Tapachula, Chiapas,
For this research,
localizada al sureste.
Our study area was the city of Tapachula, found in the southeastern Mexico.
Localizada al sureste de México, a 177 metros sobre el nivel del mar y frontera con Guatemala.
Found at 177 meters above sea level and its frontier with the country of Guatemala.
Como material biológico tenemos una cepa susceptible, Nueva Orleans, y en el mapa vemos la distribución de los 26 sitios en la ciudad.
Our biological material was composed of the susceptible mosquito strain in New Orleans and 26 field collections that are shown distributed throughout the city.
La siguiente. La recolecta de material biológico se llevó por medio de ovitrampas.
The collection of biological material of the 26 samples was done through ovitraps.
12 ovitrampas por sitio.
12 ovitraps per site.
Y del lado derecho vemos 3 ovitrampas colocadas por casa.
In the right figure, we can see that we placed 3 ovitraps per house.
En total, 4 casas por manzana.
And we selected 4 houses per block.
Siguiente. Una vez colectado el material, fue llevado al insectario del Instituto Nacional de Salud Pública.
Once the material was collected, the material was taken to the National Institute of Public Health Insectary.
Para llevar a cabo el ciclo biológico y obtener la generación F1.
Where we completed its biological cycle and obtained an F1.
Para realizar en F1 bioensayos de botellas de la CDC, por el método de la CDC.
We perform bottle assays, CDC method, on this F1.
También la detección de las 2 mutaciones involucradas en la resistencia a piretroides.
We conducted studies on the 2 mutations that are related to pyrethroid resistance.
Valina 1016 isileucina y Penilla 1534 cisteína.
Valine 1016 isoleucine, phenylalanine 1534 cysteine mutations.
Y posteriormente obtener nuestros resultados.
After this, we obtain our results.
Siguiente. Estos son nuestros primeros resultados.
These are our first results.
Expresados en razones de resistencia.
They are shown in resistant ratios.
Las líneas rojas indican la escala de resistencia.
The red lines show the resistance levels.
De 0 a 5, susceptible. De 5 a 10, moderada resistencia. Y de 10 en adelante, alta resistencia.
From 0 to 5, susceptible. From 5 to 10, moderate resistance, and 10 and higher resistance.
Y llama la atención la alta resistencia hacia los piretroides.
It is surprising that there is a very large resistance to pyrethroids.
A pesar de 5 años de uso de organofosforados, la resistencia se mantiene.
Despite the use of organophosphates in the area, the resistance has remained.
Autores indican que la resistencia hacia los piretroides se mantiene debido al uso de aerosoles comerciales.
Some authors believe that pyrethroid resistance is maintained by the use of residential insecticides.
Con clorpirifos, la resistencia es de moderada a alta.
Chlorpyrifos has a medium to high resistance ratio.
Autores empiezan a documentar la resistencia hacia clorpirifos.
Some authors have started to document this resistance too.
En Veracruz hay reporte de López encontrando en los sitios moderada, alta resistencia hacia el clorpirifo.
In the state of Veracruz, Lopez has found resistance from medium to high to chlorpyrifos in the state of Veracruz, Mexico.
Con respecto a Bendiocar, se mantienen de susceptible a moderado con algunas sitios resistentes.
In the city of Tapachula, we observe most of the populations susceptible with medium to high, with some populations from medium to high resistance.
Inmalation, a pesar de su uso, todavía se mantienen todas las poblaciones susceptibles.
And malathion, despite its use, has remained, all of its populations have remained susceptible.
Podemos observar que no hay resistencia cruzada entre organofosforados y carbamatos.
We can observe that there is no cross resistance between organophosphate and carbamates.
Esto nos hace pensar que hay otro mecanismo interviniendo en la resistencia hacia estos grupos.
This makes us think that there might be another resistant mechanisms in these groups, for these groups of insecticides.
En esta podemos ver la distribución en razones de resistencia en toda la ciudad.
In this figure, we can observe the distribution of resistance ratios throughout the city,
donde vemos esa variabilidad en los diferentes sitios.
where we observe variation throughout several sites,
Donde se ve muy clara la resistencia hacia los piretroides.
where pyrethroid resistance it's very clear.
Siguiente. Otro mecanismo involucrado en la resistencia
Another mechanism that is involved with the resistance
es la resistencia al derribo.
is the knockdown resistance.
Aquí detectamos dos genes involucrados en la resistencia hacia los piretroides.
Here we see two mutations that are related with insecticide resistance.
La bibliografía dice
Literature says
que cuando se encuentra la mutación por sí sola, la 1534, la resistencia es baja.
Literature says that when the phenylalanine 1534 mutation is found, resistance is not very high.
Y cuando están las dos presentes, se incrementa dramáticamente la resistencia.
But when they are together, phenylalanine 1534 and Solis and 1016 together, resistance increases dramatically.
Okay. Esto nos da la pauta para decir que estos genes están confiriendo resistencia a las poblaciones de Tapachula.
These findings gives us support our idea that these mutations are conferring insecticide resistance to the populations in Tapachula.
La mutación 1534 está próxima a fijarse.
And mutation 1534 is close to fixation.
Siguiente. Ok, también se buscó una correlación entre las razones de resistencia y las distancias entre los sitios.
We also look for a correlation between resistance and the distance among the populations.
Donde no encontramos correlación y encontramos una distribución espacial de la resistencia.
Where we did not find a special correlation.
Okay, para los 5 insecticidas.
For all 5 insecticides.
Siguiente. Como conclusión.
Our conclusions.
Encontramos altos niveles de resistencia hacia los piretroides.
We found high levels of resistance to pyrethroids.
Asociación de los alelos involucrados en la resistencia hacia piretroides. Associated
with alleles that provide resistance to piretroids.
Y después de 5 años, llama la atención, después de 5 años que no se aplica a piretroides, sigue persistiendo la resistencia.
Interestingly, after 5 years of the stop of usage of piretroids, we can still find the resistance.
También observamos que después de 5 años de aplicación de clorpirifos,
we observe that after the use of 5 years of chlorpyrifos,
las poblaciones empiezan a presentar de moderada a alta resistencia.
Population starts to show resistance from moderate to high.
Y completamente susceptibilidad al malathion.
And they still show complete susceptibility to malathion.
Nuestros resultados sugieren que todavía el uso de organofosforados y carbamatos.
Our research suggests that that the use of organophosphate and carbamates
son una buena opción para el control de vectores.
Remain a good option for the control of Aedes aegypti.
Y que la variación espacial encontrada.
And that the observed spatial variation
puede estar involucrada en el uso intensivo de focalizado de insecticidas.
It must likely related with the focalized use of insecticides.
Con otros factores ambientales que habría que estudiar.
With other environmental factors that we still need to study.
Okay. Siguiente, gracias.
Thank you.
Thank you very, very much for that, both of you, all of you, in terms of the in Spanish for Francisco, to Dr. Patricia for being there, and also the presentation. And a huge thanks to Dr. Saúl Lozano from the Centers of Disease Control. That is not an easy thing to do in terms of translating live. So we really appreciate that. That's brilliant. We've had quite a few questions coming through for both of you, both of the panelists. So I think we're going to, if I could ask, if I could just get everybody back together, if I'd ask Johan, Dr. Calderón, if you could come back on, and whoever's going to field the questions for the team, the Mexico, then we could then start the Q&A process. We've got quite a lot of questions that are coming through. End slide there, in terms of 5 years after the removal of the pyrethroid, you're still seeing the resistance there. Very interesting. I love the way that that's going forward to inform insecticide use and management of that. I love that, the way that you've done that is fantastic. So thank you very, very much for that. We've asked the audience to send in their questions, and they very kindly obliged. So I'm going to start off. There's a few questions for the first group, and I'll try and involve everybody as much as I can. Just bear with me 1 second while I pull this up. So from Dr. the Sheikh Anta Diop University in Dakar, in Senegal, in Africa, assistant professor and researcher there. There have been a couple of questions raised, and he's asking, I'm just pulling those up now, sorry. It's taking a bit of time to come on the screen, but we're here now. So he's asking, so this is for the first speaker, so for Dr. Calderón. Please, can you elaborate on how the discriminant dose is obtained for Chagas disease vectors and how susceptible and resistant reference strains are obtained?
Okay.
That's for you, Dr. Calderón.
Yes. Okay, if you let me, I have a small presentation about it. For I can share my screen.
Yeah, of course you can. Yeah.
Okay. Yeah. Yes.
I mean,
please, let me
Dr. Niang is asking, how can you elaborate? Yeah, the discriminant dose. Yeah, no problem. No problem. Take your time. Okay. And there's actually another question that I think he's asked. Okay. Yeah, and I suppose there's two questions there. One is from Dr. Niang, and the other one is from Gabriel Parahenao. He's asking which insecticides are used for pest management by the crop owners. So what insecticides are they currently using? And then Dr. Niang is asking, can you discriminants is obtained for Chagas disease vectors and how susceptible and resistant reference strains are obtained?
Okay, I'm going to share my screen for this because, yeah, due to the time, that kind of details, I don't share that kind of details by for explaining that with more detail. The idea with the identification of the determination of the lethal dosage was made with a bioassay that was elaborated for. Are you seeing my screen right now? Sorry.
Yeah, we can see it perfectly.
Okay.
Yeah, very clear.
Okay.
Thank you.
Our idea was to make for concentration of deltamethrin, the pure deltamethrin, and we made bioassays that were implemented and suggested for triatomins. And the idea was to use for the fifth insert. The triatomine have 5 inserts previous to the adult stage. And we use only the NINs in the 5th insert. Is the last insert previous to convert or develop in adults. We use that stage, the triatomins in that stage. We use perinins or oil pans, susceptible colony, and native pans, and we apply into each triatomine or to each insect 0.6 microliters of solution, and we determine the mortality after 72 hours. Our idea was to see after 72 hours, if we don't see any movement in the triatomins, we determine that the insect was dead. But if we see any movements in the or autonomous movement in the insects, we for that insect we think that that insect was resistant. And with those data, we made an estimation of the latent dosage, making the graphs of the dosage and the response, and we estimate those parameters. The susceptible population, the susceptible colony, was a colony that we have in our laboratory in the CIMPAT. That was collected 10 years ago in one part of Colombia, in the Casanare, that is one department in Colombia, and in the eastern plains of Colombia. And with those triatomiums, we established a colony in their laboratory, and with no any other incomes from other insecticides from the field. That was the susceptible colony. I don't know. That was the response about the methodology. What other questions?
Yeah, I thought that was a great answer and a very robust answer. So thank you very much for flashing that slide back on.
Okay.
I'm sure that Dr. Niang and yourselves can liaise with emails if there's further, if that's okay.
Yes. Any detail, please let me know.
Yeah, I'm sure that there'll be further questions. There's a very sophisticated kind of approach to it. So I'm sure there'll be other technical questions on there that we can come to directly with each other. So that's fine. Thank you for that answer. There was another point that Gabriela Parahinau made, and he was asking, did you know what insecticides were used for pest management by those crop owners in those palm plantations?
Yes, they know, because usually when we talk with the people, with the owners of the crops, they said that they don't use anything about insecticides. But we see in the field that they use, for example, malathion. Yeah, when we talk with the workers inside the crops, they admit that they use malathion, but they also know that they use pyrethroids, but they don't know exactly the concentration.
Yeah.
In Colombia, there is a management a procedure, established procedure for the managing of the pests and in ponds, and they usually prefer to use biological methods in advance, previous. But after that, we know that if the production has been compromised due to the pest, they prefer to use the insecticides, but they were very, I don't know, reluctant to say about what kind of insecticide they are using. Our idea is to try to involve to the owners and the producers of fund for helping to give us that information, and we could make some kind of collaboration with them.
This is what I wanted to ask you, actually, that given that it's a plantation, there's a private sector kind of involvement, a commercial involvement, is this a training gap issue for their workers? And if so, is there a role for yourselves to take your entomological data, which is very detailed and entomological focus, and translate that into some kind of communication training package for those workers? Is that the type of interface that you're working towards?
Yes, and that is our idea. We are trying to talk with them, but yes, usually it has been a little hard about the situation, because they are very they don't want to make the impression that probably this production could give an improvement or an increase in the transmission of the Chagas disease. But we have to see. Usually, we have to wait that.
Well, let's see. I mean, Dr. Niang's also saying that you can probably share lessons learned and how this will impact the control of Chagas disease and management of insecticide resistance, i.e. this training gap. And he's asking, do you have any community? He's actually going a step further and asking, do you have any community-based approaches for the surveillance and control of Chagas disease vectors? Is in Colombia, what's happening on a community-based approach for surveillance?
I know that based on I know, because usually I don't work directly with the health organizations in Colombia, but I know that there are some places where I see people reporting usually when they found triatomins in their homes, and when they found any insects related with Chagas triatomins in their houses. Usually they report that to the health agencies in their municipalities or in their towns. But sometimes, when my experience in the field is that we have a problem with some places where the population usually are used to live with that kind of insects, and they think that in some parts of Colombia, usually even they think that there is not so bad, because the problem with Chagas is that the mortality usually is low at the beginning of the disease, but you died some years, a lot of years after that. And they think that usually the insects are not directly related with the disease, but also is an infection related with the first phase of the disease, that usually is related with headaches and with fever and pain. That is usually the acute phase. In the places where the Chagas cases are, I don't know, a lot of different cases in one places, usually the people are more used to live with that, and they have problems to report that. And they think that usually when the problem in their hearts or in their intestine and guts related with the disease, they prefer, and they think that usually is related to other things, not to Chagas disease.
That's a fantastic answer, and I appreciate that. We're going to come back to some other questions that have come up on that. We're just moving to the other side, for the Aedes side, for a second or two, just to involve you, all of you, in this. I'll just come to that question in a second. Just bring it up. So Dr. Neil Hobbs from the Liverpool School of Tropical Medicine here in the UK, have any fitness studies been conducted on the Aedes populations? And this is to Francisco and Dr. Lozano and Rosa, Dr. Patricia Navarro. Have any fitness studies been conducted on the Aedes populations?
Hola, Pati, ¿estás ahí? I think Rosa might be having some difficulties with her audio. So I'm going to take a stab of it. We have not conducted the fitness studies with these populations. Fitness, it's hard to establish, to calculate, because it requires really long-term studies. So you have to raise lots and lots of mosquitoes in order to get a sensible measurement of fitness. What is interesting is, of course, that under the selective pressure, having these mutations that confer resistance, under this background, it's advantageous to have the mutations. They may lay less eggs or have shorter lifespans, but at the end of the day, under this background, it's advantageous.
Yeah, that's a great answer, and I appreciate that, Dr. Lozano. Thank you for that. We've got quite a few other questions coming in. So Eduardo Olmedo, can it be predicted when the Aedes aegypti will become resistant to the organophosphates and the carbamate?
Oof, that's a $64 million question. Because
in your work, the pyrethrinoid resistant thresholds, and then the differences between the carbamates, organophosphates. Is there any kind of, what's your view on that? Can it be predicted, firstly, and if so, how do you think your work can lead to that?
So from here, I have to go to population genetics, population genomics, and I can say that insecticide resistance, it's it's not a single mutation. It's it's a whole suite of mechanisms that, in the case of Aedes aegypti, they go to. The latest publication from Dr. Saavedra, she found that even changes in the cuticle can be related to insecticide resistance. if we think about this, increases in the thickness of the cuticle, or making the cuticle more less porous to the insecticide, that can lead to less contact with insecticide. So predicting those kinds of things, we don't really know what solutions nature is going to come up with. As I mentioned, it's not a single thing. There might be, big factors that could explain, a large portion of the resistance, but really there might be a lot of contributing factors to, to the single phenotype of insecticide resistance.
Yeah, that's a great answer. Dr. R.T. Rama from CARE International in India, the senior entomological officer there, has just made a point that definitely, without doubt, insecticide resistance must have its impact on physiological fitness of the vector population, and therefore must be checked following its observation and validation, just to underline everything that everybody's saying. There are other questions coming through, and I'm just pulling these through. So Dr. Audrey Lenhart, this is for Francisco. This is Dr. Audrey Lenhart from the CDC is asking, did you also observe any temporal or seasonal fluctuations in resistance patterns?
I conducted the analysis, so I comfortably can say that we did not perform that, because our for this study, we did not, our study design was not invented for temporal changes. So our sampling scheme is not that good for, for testing for temporality. So we only sample a few times a year, and not, several times a year. So we will try to look at that. That's a really interesting question, because this comes back to the question of, can we predict insecticide resistance? So definitely having an idea of how insecticide resistance fluctuates through times, it's really intriguing.
And that's a great answer as well, and a great question, great answer. Dr. Antonella Baghi-Galabupo from the University of Glasgow up in Scotland. This is for the Chagas, a question for the Chagas side of things. Hi, regarding the Chagas presentation, Antonella is asking, is the resistance detected against all pyrethroids, or would a change in one used for controlling triatamines in the housing setting, would that improve the results? Is the resistance detected against the full spectrum, all of the pyrethroid? Or if you change one, in terms of, let's say you change one that you're using to control in houses, would that improve the results?
That's a good question. We don't know. Really, we don't know about the others. That's the idea, to test the other pyrethroids are probably other kind of organophosphorates, because we know that they are using other insecticides, and the idea is to increase that. And our idea also is to see other places in Colombia with crops and with Chagas disease. The places where I checked in the last part of my presentation, that those municipalities, our idea is to see in those places we have found different scenarios, and if we see that is a common pattern that we have found. But that is the idea, to continue with that.
To continue. So hopefully there's a future collaboration with the University of Glasgow. Antonella, if you're listening, come on, and you guys in Colombia, well, there seems to be some very interesting co-interests here. So hopefully that might catalyze into some kind of research collaboration moving forward. So you never know, but there's that.
I would be happy with that.
There you go. So there's that. There are some other questions as well. Just coming back to the insecticide, so just coming back to the Aedes, I think it's for the Aedes. So Boral Chaman from the center in Cameroon is asking, from Africa, why were the insecticide doses used against the Aedes mosquitoes at very low compared to the diagnostic doses of other vectors, Anopheles, Culex? I'm not sure if I fully, because they share the same environment. So why are the insecticide doses used against Aedes set to be very low?
You know, I really don't know the history of how that dosage was set up,
Yeah.
but WHO generally has a recommendation of finding
Yeah.
DELC 90 or 99 and then
Yeah.
double that. So maybe someone in the past, somebody, came up with, this experiment, and that's where, they come up with those dosage. It just happened, might be just the biology of the mosquito that happens to be more susceptible to pyrethroids. Unfortunately, I cannot, that's just my guess. It's not my, it's not, the answer. But this is a really good question, and I would love to look for it.
Yeah, that's a great answer actually as well. So, I mean, I'm going to ask everybody if anybody has any more technical, they're quite technical, these questions, obviously, because of the nature of the session. Are you all open if people contact you all directly by email together to kind of further the discussion after the session? Are you okay with that?
I can speak for myself and definitely say yes. And I think Rosa would be happy also to have questions from anyone. And we love to share our research, I'm sure.
Brilliant. I think you deserve to get more air into this because I think it's caused a lot. People asking a lot of questions on this. Mohammed Qasim Khan from Pakistan, I think I believe he's heavily involved in the dengue effort there. I think this refers. I'm not sure if this refers. I think this refers to the Chagas because of the timing of the question. Let's see. The incriminating concentrations will be different for different genus and species. Can it vary in different regions and also may be affected with ecology? I don't know if you potentially answered that by the differences in the plantation densities and the whole link to prevalence of the disease. So maybe I'm not sure if that's to you, but that's a point he's made there. I mean, there's some interesting kind of bits and pieces coming through. That's the one there. That's it there. Yeah. Okay. 1 second. I think, Dr. Navarro is saying, yes, we can share emails as well. I believe that's what she means by that. Yes, so that's good. That's great. Affecting, just 1 second. Let me just get down into this community business. Just 1 second. Yeah, so Al-Haji, Dr. Niang is actually asking, it's kind of a mixed question, really. He's asking, are there any integrated vector management programs which go beyond the control of Chagas disease vectors and involving other VBDs in terms of mosquitoes and that kind of thing? That in Colombia, when you say integrated vector management, is it just a heavy focus on Chagas because of the endemic nature of Chagas, or is there? That's how I understand that question. I mean, I'm not sure if that's a question or a point, but I think he's asking, outside of Chagas, is it Chagas heavy or is it? I mean, I'm probably assuming, of course, it's going to involve other diseases and mosquitoes. I think that's a point, not a question. So we'll come back to that in a bit. I think that is kind of it for the questions that people are asking. Yeah, I'm just making sure I haven't missed anybody off that list. I'm sorry for the delay in my asking this. It's simply because we get a slight delay when someone asks. So that's fine. So Dr. Navarro has very kindly put her email there as well, and the panelists have agreed to kind of further the conversation moving forward. So that's good. So in terms of, we talk about communities a lot in all of this, as it were. And you mentioned these, just going back to the Chagas thing for 1 second and kind of tying it into the Aedes kind of thing. This agricultural community, this workers community, the training gaps that you've mentioned are there. Do you think these palm oil producers are ready to start funding some of this research? I mean, it's a horrible question, but all of this has to be paid for at some point to progress it, especially this your toxic detox pathway assays. I can imagine that's going to cost quite a bit to get into properly. Are they, from your opinion, when you've spoken to them, are they up for kind of financing this further? Are they open to that, or is it just a closed wall and you're just telling them entomological evidence and they're just saying yes, great, and then continuing with malathion and continuing whatever they're doing on the ground? Are they looking to change? Are they looking to fund this kind of work?
It's hard to describe that, but I think that they are interested. They are worried about that, but we are proposing for them to design some strategy between agriculture and health, trying to make a pest control that usually consider the control also of triatomins, and try to, if they are focused on the control of pests, they also include the control of triatomins to avoiding the transmission of the migration of triatomins to the houses. But until now, until this moment, the answer has been very. They prefer not to talk about it, and probably we need to try to have more communication with them. But yes, we don't have any conversation about these two sectors right now, and usually they said that, and they are worried because they consider that sometimes the crops are some places that have forests in some regions of Colombia, and they help to the conservation of the forest because when you see a oil palm crop, usually is a big forest. It's a big forest of one type of palms, and they think that this is a conserved area. But with the doubts of the problem that supposition could have, but with the next step that probably created or made the possibility that the production of oil could increase the transmission of Chagas, we haven't had a good communication until now. Probably in the future.
This is what I was about to ask you. You've done the hard work. You've done the hard work. Isn't it now turning into a communication challenge? That was exactly what I was about to ask you. And if it is, what are your plans around that? Because there is an issue, there is a linkage. You're showing that directly, and I'm sure if you map it out, the further studies outside of the two areas you've mentioned into other regions, you'll see even stronger correlation. So the case is there, the evidence is building. What are your communication plans around that to harness this kind of, it's the commercial sector, and if they're involved in driving some of this resistance, and as you've shown, what are your communication strategies around that, if you don't mind me asking?
No problem. We have no very developed communication strategies with that kind of sector. But I think that we have initially with the publication, we think that we are going to have any type of communication between sectors, but no, that the communication was not developed. And I think that right now we are trying to Camila Gonzalez, that is the director of my research group. She are going to talk with some people working in the palm sector, trying to show that and trying to see if they are interested in developing a strategy. But until now, until my knowledge, I think that we have to wait. And I don't know if they are waiting more cases or more evidence, but I think that we could try to seek new evidence with them. And see if the problem probably is only focused on one part of Colombia, there is no problem. But if we found the problem in other places, we can make a common strategy.
Yeah, that's a great answer. I'll be really interested to see how that unfolds moving forward. So that's actually great. Just turn to the Aedes aegypti resistance team there. I just wanted to let you know that in terms of dengue, at least, we ourselves run the World Dengue Day campaign, where we're asking the United Nations and WHO to ratify an official World Dengue Day. We had kind of the soft launch. We've got over 30,000 signatures on a petition that's out there in the world, from right across the world. We've held many kind of events and kind of panels and discussions to push that forward. We'd love to involve you in that, in our dengue kind of group in that. And if you're up for it, I'll send you an email after. You can see what we've done and who we're working with. I think you'll really enjoy that. And secondly, probably very interestingly, this morning, I don't know if you saw the session, the opening session, but with Dr. Roman Valiuduan from the WHO, but there was a call to arms because the WHO have started an arbovirus group. They're trying to form an embryonic kind of pulling together lots of people. There's a call to arms for people to get in touch with them with regards to that, Aedes and all the diseases around that. So I don't know if you'd be up for kind of contacting him around that. If you need any kind of help to get through to him on that, let us know. But they're looking for people probably working in exactly this particular area. So I think that would be great. That was in the first session this morning, especially for your work. All of that said and done, I think we're coming towards the end. There's been one more question by Boral Charman to Dr. Rosa. Please, have you explored the possible origin of that resistance observed in Aedes populations there? And that's kind of straight to the core.
Yeah.
Might as well throw it.
That's an amazing question, and that will take us back to dancing, swing, and go-go, and even further back. Because there was malaria and dengue and yellow fever in the United States, and then World War II happened, DDT appeared, and that provide countries with the ability to start to treat with insecticides large areas. So, where this mutation came from, it could have been incipient in the populations. It was already there. It was just a matter of putting pressure on the mosquito populations in order to emerge. Could it have appeared de novo? That's the probability, If the question is more regarding on this particular city, where did it came from for this particular city, Aedes aegypti, it's a world traveler, and it moves from one place to another, and that's one of the risks with Aedes aegypti, that once a mutation, just like bacteria, once a mutation gets established in the population, in order to revert back, it takes again, it can take thousands and thousands of generations. So it's an amazing question, and I actually it would be in the best interest of the audience to just look at the history of DDT. It's an amazing history.
Yeah, exactly. That's a great answer. That's a great answer to that question. So thank you for that again. So I think that's kind of taken us towards the end of this session. I'd like to say an absolutely massive thank you to all of the panelists who have made time today to kind of sit there, to Francisco, to Rosa, a special thank you to you, Saúl, to Dr. Lozano for actually translating and everything. Real time is not an easy thing to do. We really appreciate that. So thank you for that. And to Johan, to Dr. Calderón, in terms of joining us from Colombia and kind of looking at that interface. I thought that was both were fascinating. And as you can see, the audience are really getting into the questions. And a round of applause for all of you, and a round of applause to the wonderful audience. I believe we have a final session that's coming up now. My colleague Marianne, the other co-founder, has very kindly put up the next link. Each of the rooms we go to has its own link, so it's not confusing. It's just the way this platform is set up. So I'd actually really urge all of you to make that hop onto that. And you'll see my face again. And I will apologize now, because on every session I have to ask everybody, please put your name, country, affiliation in, because it wipes it, because it's a different room. So I'll have to ask you to do that again once we get there. I think that would be great. I really hope, because of the international flavor of the panel and the actual the audience, that something interesting comes out of this for future collaborations. You all deserve it. And I think there's a tremendous amount of interest in your work. You can see that from the questions coming through. So that's brilliant. So a round of applause to all of you. Thank you very, very much. I am not very good at saying goodbye, so I'm going to so I just wanted to say thank you. Please do join us for the final session. we're actually looking at leveraging communities for vector control, and a kind of a range. We have speakers from Kenya, speakers from Italy, and speakers from Berkeley in California, Berkeley, the University of California, looking at various approaches, art, science, social media, apps, actual hardware, all sorts of stuff. So please have a look. We're going to be jumping over to that session, and I believe it starts in about 15 minutes from now. So thank you very, very much, and I'm going to have to end the session here. I hope we can continue this conversation moving forward. All right. Thanks a lot. Everybody saying thank you. Extraordinary sessions from everybody. Everybody saying thank you from the panel. Dr. Rama from India is saying that as well. So thanks a lot, guys. All right. Yeah.
Thank you. Have a nice day.
Dr. Rosa as well, Dr. Navarro, take care. All right. Bye-bye, everybody. Take care. Bye-bye.
Bye-bye.